the binding of this protein to the DNA may influence variation in DNA methylation. And what Dirk Schubler's work has shown is that variation, genomic variation, so genetic variation that influences the sequence at the binding site, can also predict variation in DNA methylation. So just in the last few minutes, I'd like to tell you a little bit more about the work that we've been doing on weighted gene co-expression network analysis. So as I mentioned, this is a form of analysis that looks at the correlational structure of DNA methylation. So here you're trying to identify co-methylated CPGs. So again, we're just running this on variable CPGs, so CPGs that show inter-individual variation. And we're asking whether or not we can classify these CPGs into various different networks. So what we did was ran the WGCNA, so the analysis, only in those individuals with a history of child abuse and neglect, given the marked effects of child abuse and neglect on variation in DNA methylation within the nurse-family partnership. So here we're just asking, what are the modules that are evident in those the individuals with a history of child abuse and neglect? In the next step of the analysis, we said, okay, we've identified these networks in those individuals with a history of child abuse and neglect. Can we identify the same networks in control individuals, in individuals that don't have a history of child abuse and neglect? And that's an analysis called module preservation, where you're basically trying to identify networks in one group and test whether or not they are present in your control group. And what we identified was 10 modules that showed evidence for weak preservation across the groups. So with weighted gene co-expression network analysis, every module gets a really fancy color name. So that's what you're seeing in this correlation table up here. You're looking at the correlation between these modules that show weak evidence of preservation across groups. So modules that seem to be more enriched in those with people with a history of child abuse and neglect. And you're looking at the correlation of those networks with symptoms, current symptoms of PTSD, with PTSD diagnosis in this particular case. And what we found was that there was 2 modules that were significantly associated with symptoms or a diagnosis of PTSD at age 27 years of age. And with one of these modules, Blue 2 as it's called, we ran an enrichment analysis. And again, what we found was there seemed to be an enrichment of processes related to transcription factor binding, so proteins binding nucleic acids, binding DNA. So again, I think this is fairly consistent with what we saw with our linear regression analysis, showing an enrichment of the glucocorticoid receptor, estrogen receptor, and progesterone receptor. So again, pointing to this mechanism that transcription factors binding DNA may influence variation in DNA methylation. But importantly, with the symptoms available on PTSD, these may be also related to the presence or absence of psychiatric disorder. So just to conclude, I think with these studies, we've been able to demonstrate with, for example, the MAVEN study, that the early care environment does seem to play a role in shaping the developing methylome. It does associate with variation in DNA methylation. And there does seem to be significant overlap with those sites that are also associated with child mental health. Now, one of the reasons that I'm particularly interested in the infant attachment data is because there's at least 2 studies that show that the attainment of a secure attachment style can buffer the effects of adversity experienced earlier in life. So I think perhaps what is especially interesting about this methylation data is asking the question whether or not we can see a molecular correlate of this social buffer, of the buffering capacity of infant attachment. And obviously that will require further work. But as we noticed from the nurse-family partnership data, we see really quite widespread effects of child maltreatment on methylome variation. But the question is whether or not this plays a causal role in health and disease. Because as I mentioned, we saw quite a marked effect of lifestyle factors. And we know that there was quite a long time from the termination of the program until the biological sampling. So trying to identify or do a causal analysis is very difficult, near impossible, with these cross-sectional data. But I do think that they provide proof of principle that both potentially early intervention, but also early adversity, play a role in shaping methylome variation. So if we think about the methylome that I've just talked about, this genome-wide DNA methylation, of course, we know that underlying the methylome is the genome. And although I haven't spent very much time on it today, we do know that genetic variation is very important for variation in DNA methylation. So this is a very important factor to consider. And in all of the studies that I've mentioned, we also have genome-wide data on single nucleotide polymorphisms. So we're incorporating them into all of our current models. But I think what we've been able to see from these data is potentially there may be a role for early intervention to influence variation in DNA methylation. And as I mentioned, we also see an association with early adversity such as child abuse and neglect. But the question is then trying to discern the effects of child abuse and neglect from the sequela of child abuse and neglect. But maybe that's the wrong question. Maybe we want to actually identify the cumulative effects of these developmental pathways that are associated with either an adverse early rearing environment or a positive early rearing environment. And I think ultimately identifying those areas in the methylome that intersect, where you see the interaction between the environment and the genome to predict variation in methylation, might be most informative for identifying those sites that are causally associated with both health, disease, resilience, and comorbidity. So I'd just like to finish up there and thank, of course, my mentor, Michael Meany, and all of the team at the Douglas. Michael Kobor, who has run our 450K arrays for us. He's an excellent collaborator. Joanna Holbrook and the team in Singapore, and of course, Vivek Lover and Tom O'Connor, who I worked with on the ALSPAC quarter. And of course, David Oles, who's provided us access to the nurse-family partnership, which has been a really interesting form of analysis for us. So thank you very much for your attention.
I think we have time for a question or two.
Oh, oh good. I'm glad Michael's not asking the question.
This is, I guess it's kind of a comment, really. You presented some very small correlations with PTSD. And I think those are actually more impressive than you may even realize. First, that you got them. But also, PTSD is incredibly noisy. You're looking at something that's actually in the brain versus this category that is just really sloppy. You know, it's a diagnostic category, and there's imprecision. But also, there's tremendous heterogeneity in that diagnosis. One of my former students, Isaac Elter-Levy, published a paper last year called 630,129 Ways to Have PTSD. And it's actually, what he did was he just did a statistical analysis of the possible symptom combinations that you can have and still have PTSD. And there are literally 630,129 symptom combinations. So with that kind of heterogeneity, the fact that you got even any correlation is remarkable. So there's something there, definitely.
Yeah, well, thank you very much for that. And you know, I couldn't agree more, not only with regard to I didn't realize there was that much heterogeneity in PTSD, but also one of the limitations of the study is that there's also heterogeneity in, you know, child abuse and neglect classifications. You know, they're all from Child Protective Services, but, you know, I haven't stratified in terms of the type or, as we heard yesterday, the timing of these exposures. So I think that might help to resolve some of these associations as well. But, you know, that's something that definitely I'd be interested in looking into more.
You can give the quick answer now, but I'd love the expanded answer during a break or something. I do research in shelters for homeless families. Those families are extremely interested in the issues that we're talking about at this conference. The mothers are worried about the stress that they're undergoing and its impact on children. And although many of them don't have a great education, they see TV, they know that this kind of research is going on, and they're concerned. They're also extremely willing to participate in research. And when they're in mid-crisis, basically, because they're in an emergency shelter for different reasons, but often there's either domestic violence or a sudden crisis in a family that's already very disadvantaged. Given what you've studied so far, what kind of research would you want to do with these families who are very willing to partner with you, who are in the middle, the adversity is ongoing?
Yeah,
right now, a lot of these mothers are pregnant, they're young, homeless parents tend to be very young. What kind of work would be illuminating given that kind of context?
Yeah, well, obviously, you know, I'm completely biased. I guess one point to make, though, when you mentioned about these families, you know, seeing stories in the media and stuff, and this is something that I was particularly sensitive to during my PhD working on maternal prenatal anxiety and looking at the effects on child outcome. So you're basically saying, you know, prenatal anxiety predicts child mental health, and then you're telling women, don't get anxious about being anxious, you know? So it's a bit of a paradox. But, you know, the thing that I would say to those families is that most children aren't affected. And, you know, even if we see the graph that I showed in terms of PTSD diagnosis, you can see that even in those individuals that have been you know, exposed to child abuse and neglect, you still see that there's quite a large number of individuals that don't have that diagnosis of PTSD. I can't comment on what other impairments or adaptations they may have, but you can see that you know, a lot of them don't have this diagnosis. In terms of what I would actually like to get from these families is that really, I think collecting biological samples is going to be very helpful. And one of the issues that we're coming up against with these analyses, where we're increasing the number of sites that we look at across the genome, is power. And it's a criticism of this work is trying to get an adequate sample size, and also adequate phenotyping, you know, making sure that we do a good job of characterizing the environments that these individuals are coming from. But yeah, I'd like to talk to you more about how to expand on that answer.
Yes, Chris.
Thank you for a very clear and concise presentation. I've been to a number of talks where the genetic information hasn't been presented in such a way that it could be understood so easily. Thank you for that. Actually, working with the Nurse Family Partnership in the past, and also currently running a clinic for 0 to 5-year-olds that have suffered from trauma or problems in attachment, I find the information especially compelling. And in looking at 27-year-olds, if you could look at any age group, whether it be 10 or 20 or whatever, and knowing that David Olds could possibly give you an opportunity to look at a younger age and another population, what age would you pick, and what would you hope to find?
Yeah, well, so to be honest, I would try and go the earliest that we could go. You know, one of the, I know this might sound a bit far-fetched, but one of the aspects of my PhD work was working on the placenta and looking at potential placental mechanisms that are sensitive to the in utero environment. And in fact, Michael and the team in Singapore have some very interesting data on umbilical cord, for example, showing that there's variation in DNA methylation that arises as a function of environmental factors interacting with the child genome to predict DNA methylation. So to be honest, the first tissue that I would want is the placenta from those women that have been exposed to a perinatal intervention, and then potentially buccal epithelial samples over the first year of life or until the termination of the program at age 2, to really see if the intervention is associating with the maturation of the methylome. So we know that there's variation in the methylome that occurs over the first 18 months of life. And the data, this gives me an opportunity to show the slides that I skipped over. Hang on one second. So we know that there's variation in the maturation of the methylome. So here what you're looking at is a longitudinal study of a small group of females, and they collected repeated blood samples at early in life and then up until the age of 5 years of age. And what I think you can appreciate from this graph is that at birth, all of these all of the samples taken at the birth time point clustered together. But I think what you can appreciate as you look at the other time points at the 2.5 years and at the 5 years, what you begin to see is that there's some kids who are aged 2.5 years, but they're looking more like 5 years. And there's some kids that are 5 years that are actually clustering closer to the 2.5 years of age. So I think what you're seeing here is that you're getting differences, inter-individual differences in the maturation of the methylome. And I think this could be very interesting to explore in terms of the different variations in the early care environment or treatment group as a predictor of this methylome maturation.
Well, and certainly it would eliminate all the complications of the environmental experiences following the termination of the program at 2.
Exactly, yeah.
Victor, quickly, yes, one last question.
Yeah, I think you just addressed this, but what change do you expect to see in an effective intervention? Is it the prevention of methylation, or is it a change down the chain?
Yeah, so that's the big question, you know, that hopefully we'll be able to address with, you know, studies in collaboration with Chad and Jenny and others, where, you know, we try to do this in a prospective manner to identify what are the treatment effects on variation in DNA methylation, or are there treatment effects on variation in DNA methylation, or is it the fact that by preventing child abuse and neglect, that this is what's influencing the variation in DNA methylation? So I think it's still a bit of an open question. And of course, it will also potentially depend on the tissue type that you're looking at.
Thanks.
Great. Thank you, Kieran, very much. Wonderful presentation. They are switching out computers. That'll give me an opportunity to introduce our next speaker. Dr. Bekh Bradley-Davino received his PhD in clinical community psychology from the University of South Carolina. As a clinical person myself, I'm actually very excited to hear about the translational aspects of his work. He is currently associate professor in the Emory University Department of Psychiatry and Behavioral Sciences and serves as chief of the mental health service line at the Atlanta Veterans Affairs Medical Center. There he specializes in the etiology and treatment of post-traumatic stress disorder, or PTSD. Dr. Bradley-Davino brings significant experience in clinically and empirically grounded research focused on the identification of risk and resiliency factors in response to lifetime stress and trauma exposure. This work includes examination of both environmental and genetic determinants of the risk for PTSD and other forms of psychopathology. He is currently the PI on an NICHD-funded R01 proposal entitled Mechanisms of Intergenerational Impact of Maternal Trauma. This grant will identify how trauma exposure in one generation can affect offspring in a manner that increases the risk for various health outcomes across development. While his work has clear implications for many different traumatized populations, Dr. Bradley-Davino's work on the intergenerational effects of trauma will facilitate a better understanding of the risk for mental health outcomes across generations in the child maltreatment population, identifying relevant mechanisms to be targeted in clinical intervention. So with that, I'd like to bring up Dr. Bradley-Davino.